Hello and welcome. Today we're going to take a really close look at the remarkable journey of thyroid hormones. We're going to follow the path right from a single humble atom of iodine and trace it all the way to the powerful genetic commands it issues deep inside our cells. Right then, let's get started.
So it really all comes down to this one central question, doesn't it? How does a simple element, iodine, something we get from our diet, get transformed into one of the most sophisticated hormones in the body? A hormone that literally sets the pace for our entire metabolism. Well, that's the fascinating process we're about to unpack.
You know, our story doesn't actually start inside the thyroid gland. It starts out in the bloodstream. This is where the gland's functional cells, the thyrocytes, have to go on a hunt. They have to actively capture their one essential raw material, iodide, from the plasma. If this first step fails, the whole production line simply grinds to a halt.
And that brings us to our first critical phase, the iodide trapping mechanism. This isn't a passive process at all. It's an incredibly efficient, active system where the thyroid gland secures the iodide it needs, laying the absolute foundation for all the hormone synthesis that follows.
The star player here is a protein called the sodium iodide symporter, or NIS for short. It sits on the basolateral membrane of the thyrocyte. That's the side facing the blood. Think of it as a highly specific molecular pump, a gatekeeper that performs the crucial task of actively pulling iodide into the cell.
So how does it manage to concentrate iodide against what is a very steep gradient? Well, it's a beautiful example of secondary active transport. NIS cleverly hijacks the powerful electrochemical gradient for sodium, a gradient that's constantly maintained by the cell's sodium-potassium pump. First, two sodium ions bind to the outside of the NIS protein. This binding action makes the protein suddenly very interested in iodide, dramatically increasing its affinity. Once the iodide is on board, it triggers a conformational change. The whole protein flips, and it deposits all three ions inside the cell. Clever, eh?
And the result of this mechanism is just staggering. The thyroid can achieve an intracellular iodide concentration that's anywhere from 20 to 50 times higher than what's in the blood plasma. And in a hyperactive state, say in Graves' disease, that ratio can shoot up to over 100 to 1. This massive accumulation is, of course, absolutely essential for synthesis to even begin.
Okay, so now that the iodide is trapped inside the cell, it needs to get to the factory floor. It's transported across the apical membrane and into the follicular lumen. And it's here that we enter what you can really think of as the hormone assembly line.
The foundation of this whole assembly line, the molecular workbench, if you will, is thyroglobulin, or Tg. This is a colossal glycoprotein, a huge scaffold. Without this structure, hormone synthesis would be completely disorganized. And this has real consequences. If thyroglobulin is mutated so it can't be iodinated properly, the entire process fails, which can lead to a condition called dyshormonogenesis.
Right, so if thyroglobulin is the workbench, then thyroperoxidase, or TPO, is the master craftsman. This enzyme is positioned perfectly at the apical membrane, right where the action is. It meticulously orchestrates the entire chemical construction of the hormones, and it uses hydrogen peroxide as an essential cofactor to get the job done.
TPO's job is threefold, and each step is absolutely critical. First, it has to oxidize the iodide, making it chemically activated and ready to react. Second, in a process we call organification, it attaches this reactive iodine onto specific tyrosine residues on that thyroglobulin scaffold. And finally, it catalyzes the coupling reaction, joining these newly iodinated tyrosines together to create the final hormone structures.
So, that organification step creates our fundamental building blocks. If you attach one iodine atom to a tyrosine, you get monoiodotyrosine, or MIT. Attach two, and you get diiodotyrosine, or DIT. These are the precursors, still physically part of that enormous thyroglobulin protein.
And now for the final bit of chemical magic, the coupling reaction, which is also catalyzed by TPO. It's quite simple, really. Join one MIT and one DIT, and you form T3, triiodothyronine. Couple two DIT molecules together, and you get T4, thyroxine. But what's so crucial to remember is that all of this is happening while these brand new hormone molecules are still physically attached to that thyroglobulin backbone.
So we now have our finished hormones, but they're locked away in the colloid, still bound to thyroglobulin. So the next phase is all about packaging and distribution. It's about liberating these hormones and then shipping them out to every single cell in the body.
The release process is really quite elegant. The thyrocyte literally reaches out into the lumen and gulps down droplets of colloid through endocytosis.
This little vesicle then merges with a lysosome, which is essentially the cell's recycling center packed with digestive enzymes. These enzymes then chop up the thyroglobulin scaffold, and that liberates the free T3 and T4, which can then simply diffuse out of the cell and into the bloodstream.
Now, as soon as they hit the blood, something very important happens. The vast majority of T3 and T4, we're talking well over 99.5%, immediately gets grabbed by transport proteins. The main one is called thyroxine-binding globulin, or TBG. This is vital because it creates a large, stable reservoir of hormone in the circulation. This leaves only a tiny fraction, less than 0.5%, that is actually free and biologically active.
And this brings us to an absolutely crucial point for clinical practice. It's only the free, unbound hormone that can actually get inside a target cell and have an effect. The rest is just an inactive reservoir. That's why measuring the free T4 level is the best indicator of a patient's true thyroid status. Measuring the total T4 can be misleading because it can be affected by changes in binding protein levels, something you might see in pregnancy, for example.
Right, we've reached the climax of our journey. The free hormone has traveled through the blood and arrived at its target cell. Now we're going to see how it delivers its final command, not at the cell surface, but by going deep inside the nucleus to interact directly with our DNA.
Once inside the cell, another important distinction becomes clear. T4 is mostly just a pro-hormone. Our peripheral tissues are full of enzymes called deiodinases, which are brilliant at clipping one iodine off T4 to convert it into T3. And this is a key control point because T3 has a much higher binding affinity for the thyroid hormone receptor. It really is the primary, more potent, active form of the hormone.
So let's look at this mechanism of action. It's wonderfully elegant. The thyroid receptor, or TR, doesn't just float around. It's already sitting on the DNA at specific sites, often paired up with another receptor called RXR. Now, when there's no hormone around, this complex actually recruits co-repressor proteins, which actively silence the gene. But when T3 arrives and binds to the receptor, it triggers a conformational change that kicks the co-repressor out and recruits co-activator proteins instead. This flips the molecular switch to on, and transcription of the gene begins. This direct control over our genes explains why thyroid hormones have such a slow onset but a very long-lasting sustained effect on our metabolism.
So there we have it. We've gone from a single iodide ion all the way to the complex regulation of our own genome. For the last part, let's explicitly tie all of these intricate biochemical steps back to why they matter so much in a clinical setting.
You see, understanding this pathway isn't just an academic exercise. It's fundamental to medicine. A genetic defect in the NIS pump means you can't trap iodide, causing congenital hypothyroidism. The TPO enzyme, it's the target for the drugs we use to treat an overactive thyroid. And it's the primary target of the autoimmune attack in Hashimoto's disease. We've already mentioned how misfolded thyroglobulin causes disease. And of course, knowing the difference between free and bound hormone is paramount for interpreting blood tests correctly. Every single step we've discussed has a direct clinical correlate.
And that leaves us with a final thought to ponder. We've just walked through an incredibly elegant, multi-step biological process, but it's also a remarkably fragile one. Its utter dependence on a single trace element from our diet and its vulnerability to autoimmune attack should really tell us something profound about the delicate biochemical balance that is required day in and day out to maintain our metabolic health. Thank you for joining me.